Okay, I'm going to, 1 second. Okay, so I will share my screen. Okay, now you can see my screen, right?
Yes.
Perfect.
Yeah.
Perfect. Okay. So yes, just the main important thing that I want you to remind from the previous day is that we have multiple ways of representing the biological information because there are multiple processes. So mainly what we are going to see today is sequence alignment. That can be between DNA or between proteins normally, okay? So, as you remember from the previous session, we have DNA, that DNA is stored in the forward sequence that goes from the 5th end to the 3rd end. That the name is because of the carbon, the ends carbon of the sequence. That we also have another strand for the DNA that is the reverse strand, that from that strand we transcript to RNA, that is another molecule that will be translated also to proteins. So we have those three main components in the biological information, let's say, that are DNA, RNA, and proteins, and that we can represent them by sequences of letters in bioinformatics, and we just store those long chains of letters that in the case of DNA and RNA represent nucleotides, okay, and can be A, C, G, and T in the case of DNA, and U for RNA. And then we have proteins that are composed by amino acids, and also we have 20 essential amino acids that are represented also by letters, okay? So when we say that we want to make, for example, alignments, is just to compare two or more sequences of generally DNA or proteins and see which letters are matching, what in DNA will mean nucleotides, or also base pairs that are almost the same thing. We name them independently, okay? And in proteins would be amino acids, okay? In the slide that you are seeing, we can also find other information, not just sequences, also gene expression that is found from RNA. Gene expression is the part of the bioinformatics that are in charge of analyzing in some conditions or some tissues or under some diseases, for example, different samples, how many copies of RNA do they have? Because sometimes there will be more than the base level, that we will say that the gene is overexpressed, or there will be less, that we will say that the gene is underexpressed, okay? Same thing with proteins. We can see also the expression of proteins. There are also databases that give us information about the protein function annotations or the protein-protein interactions, also protein DNA. It can be those regulation factors that we talked the previous day. And that will be a very summarized schema, but we can understand everything more or less with this. So there are some very important biology databases. There are tons of biology databases nowadays because it is a very increasing field, and the technologies are getting better and better. So we have very different and very large databases regarding biology. Okay, for I don't know what is itself going forward. Okay, for nucleotides, there are three main databases. That would be the NCBY from the USA, MBL from Europe, and DDBJ from Japan. Okay, we normally use these both. The NCBY has most of everything, but also MBL. Okay, and there are also databases that are more focused on some genomes of particular organisms. Can be fly-based, that is for Drosophila. Drosophila is a fly. I don't know if you are common with that, but it's like a model organism where most studies are being performed. As for example, in plants, we have Arabidopsis. Also, we have SGT for yeast, or ensemble for human, mouse, and there are also other animals. But that is when we are looking for the complete genome. When we are looking for a specific, for example, gene, or maybe a transcript or something more specific, we go to nucleotide sequence, although they also have for multiple organisms, but they are organized as entries are genes, for example. Okay. Also, we have proteins databases as UniProt, SwissProt, PDB, MDB, Scope. I think Alejandro already introduced you some of those in the first day presentation or the second day presentation, but just to remark that there are a lot, okay? And I don't know what is passing, say, okay. And that for one task, it will be most useful one database, and for other one, it will be much more useful another. So we have to keep that in mind. Also for bibliography, we have PubMed, that is being the most mined source to extract biomedical knowledge, for example. And also for metabolic pathways, we have KEGG. Another one would be Wikipathways, for example. Plenty of them. Here I wanted to show you some important file formats, because how do we store those sequences of DNA or RNA or proteins? There are some concentrated file formats that are... Give me a second, because I don't know why it's just passing like from one to another. Sorry. Okay, I think I have. Okay, as I was saying, we have different format files. The most important one is the FASTA file. I don't know if you are familiar with it, but those files that I uploaded in Moodle were FASTA, and FASTA is just as simple as a header that is preceded from a major sign, okay? And then the second line, it is the sequence. So in the header, we store which gene or which protein we are talking about, and then in the next lines we store the sequence itself. We can have in a same FASTA different different entries. So the file will have several major signs, okay, that will correspond with its different entry. So just for you to see, open please the file that I uploaded, the first one, well, I don't know if it is the first one, but the one that is called TC573649. This is a gene from rats, okay, from a rat. So as you can see, the first line is the corresponding with the header that is preceded for major sign, and then here we would have stored whatever, okay. If we can say rat gene, and
excuse me.
Yes.
You can just make a little zoom on.
Yes, maybe. Of course.
Yes, thank you.
Here, you can see it better now.
Yes, it's better.
Okay, perfect.
Yes, thank you.
Nothing. Yeah, you can write wherever you want to identify that the sequence below is that gene or that protein. Here, as you can see, we have a DNA sequence because we have G, T, A and C, okay? We don't have amino acids. And most of the time, RNA is relegated to specific things. We normally work with DNA. Okay, I remind you that this sequence would be a forward string. So here we would have the fifth end, and here the third end. This is important because when we want to align two sequences, they have to be both 5 to 3. If not, they would be reversed, so the alignment would be completely opposite. Also, we can store in a same FASTA, as I told you, different genes. For example, this would be the same protein, the protein that, one protein that is codified, that is the fractaxin, that is involved in how do you say in English? Hierro. I don't know the word in English now. Oh my God.
Carlos can translate if you
hierro?
want.
Carlos, do you know how to say hierro? That I'm completely forgot. Carlos, are you here?
Sorry, yeah.
Oh, do you know how do you say hierro in English? That I forgot. Hierro.
Sorry, can you repeat it again, please? That my internet so it's having problems. Sorry.
How do you say hierro in English?
Iron.
What? Sorry.
Iron.
Iron.
Yeah. Sorry, my fault. Okay. So this is a protein that is in charge of metabolizing iron in cells, and it is very conserved along species. So you have the same protein, but in different species. In the first entry, you have, for example, for mouse, then for rats, for dogs. Okay, so each major sign will represent a different entry with its different heading. Yeah, and you can have as many as you want. These types of FASTA files are called multi-FASTA because they have multiple entries. Okay, so I again repeat. Here you have a DNA sequence for one gene, a rat gene. This is another completely different gene. Okay, this one we are going to make some alignment, and with the proteins we will make others. Okay, so I am going to put the original header, and let's proceed with the presentation. We have also SAM files. SAM files are sequence alignment maps. Those are for when you are sequencing a very huge genome, for example, you have to split all the DNA in fragments that are not as long as the whole genome. So some files are helping us to structure that that alignment, but here I want you to differentiate from the alignment that we are going to see today, that is alignment of sequences, than alignment of reads. As I will explain you later, reads are those fragments of DNA that are stored when we are sequencing, okay, of next generation sequencing, that are those modern techniques that are used to sequence and that are very fast, and you can sequence very, very huge genome, for example, okay? Also, BAM files. BAM files are the same as SAM, but are compressed in a binary format, okay? Also, some have different sections as FASTA-read. They have relevant information about those alignments of reads. I skipped a FASTA-Q. FASTA-Q is another type of FASTA that has some more information than FASTA, because was developed by the Sanger Institute, that is an organization that developed some techniques for sequencing, and the Q stands for a score that they give for the threshold of certainty of how that sequencing was done, okay? But as I said, the most used is FASTA, and FASTA are used for almost everything, okay? Also, we have a variant calling format, that those are when we are analyzing the same genome in different individuals, so we can see the variability in a population, the SNPs that we call. I don't know if you are familiar with that are only some nucleotides that are modified from the entire genome, and that give that variability to the population. Also, general feature finding format, that is for keeping information about gene structures, most of all. Also, gene transfer format is quite similar to GFF, and also is to describe features and structures of genes, DNA and RNA, but also some types of proteins. Okay, here is the same thing that we have just shown in the other file. Here you have again a multi-FASTA, because we have different entries that I'm pretty sure that are different genes. They don't have to be compulsorily genes, okay? They can be just a segment or more than a gene that we want to analyze to make alignments, or to search for patterns or whatever. I didn't say it, but the files that I provide you are all file.fasta, but we can also keep those files as file.fa or file.fsa, okay? Some of the most common tools for bioinformatics will just recognize those FASTA files. I don't have anything installed in my local computer, so that's why they are just like text files, okay? But for example, some softwares, as it could be Muscle or I don't know, some phylogenetical analysis tools, they will automatically recognize those FASTAs. Yes, once again, just remind you that we have the header and the sequence, and anything else that is not like that. I mean, if we put, for example, dot comma, the algorithms or the programs that are using FASTA files won't take care of those lines. Okay, it's like if we put a hashtag for a comment in Python, let's say, that is with a dot comma. Okay. We are going now to start thinking of alignment, of sequence alignment. The biological significance of sequence alignment comes from the fact that DNA and protein sequences define the protein functions, or all the functions really in living beings. Okay. So the more similar two sequences are, the more similar the functions that are encoded by them will be. The sequence of a same gene in a set of species, as we saw before, they were a protein, but imagine we had that protein, instead of protein being a gene for mouse, rat, human, whatever, different species. The more different those sets of genes of different species are, the more different are in the phylogenetic tree. The more distance the two species are in a phylogenetic way. Okay, I didn't say it, but phylogenetic means how close two species are based on the distance in the DNA or protein sequences. So normally when we align two sequences, they have a high similarity, and they have a high similarity, it is because they are homologous. What it means that they have a common ancestors. Okay, an specification event occurred and they no longer are the same species, but in a past they were. Okay, so I already mentioned them, but there are two important concepts here that will be similarity and homology. Okay, while similarity is a more quantitative term, two sequences are or are not homologous, okay, as they derive or not from the same common ancestor. From the similarity we can infer homology, so having a measure of similarity will let us think that some two sequences are homologous or are not. The accumulation of mutation in the DNA during time is the cause that two sequences are from different species. So they have some changes between species, but the most close those sequences are, the most close those species will be. So what do we want when we align sequences? We want to measure that similarity that we are saying, that is a quantity, and from that similarity infer if the two sequences are homologous or are not. We also want to determine the correspondences residue by residue, okay? Find functional dominance, for example, or compare a gene and its product, or search also for these evolution relationships which positions in the sequence are homologous of other sequence that will tell us in the final instance which regions are more conserved among species. For example, if some kind of proteins are really needed for life, as we saw with exokinase the last day, that is very conserved along the species because it is essential for glucolysis. That is one of the main pathways in the metabolism of all of the species. So yes, just to remark that an alignment provides an hypothesis of how the divergence between two species may have occurred. In the case of DNA, we make alignments to identify genes. If we, for example, have a sequence, a very long chain of DNA from whatever environment, and we want to see which genes we have in that sequence, we will align it to other sequences in different databases to search if we have correspondences between other genes in the same species or in other species. Also to see which regions are the codifying regions, the exons that we saw the last session, and also to give the sequence what we call expressed sequence tags, ESTs. We also, as I already told you, make studies of population variability with the SNPs, those single nucleotides polymorphisms that will be the origin of variability between different individuals in the same population. For example, humans, all the people have different kinds of SNPs. Well, not all. Sometimes SNPs also are related to some diseases, but they are very little mutations that can lead to slightly different phenotypes. Okay. Also, we can compare those non-coding sequences that were most of our regulatory sequences, what we call introns. Okay. In the case of proteins, we can use sequence alignment to know the general function of that protein. If a protein is very similar to another protein, it will be very probable that those proteins have some functions in common, okay? Because as the sequence in the protein will lead to the dominial different catalytic residues, okay? If we have two very similar sequences, the domains that we are obtaining will be the same. Also, we can model the three-dimensional structure of proteins, how they make the connection between the amino acids, if they are in an helix or if they are how do you say in English, beta lamin, or whatever structure they have that is important also for the things of the interactions, where they are binding, or to which proteins or DNA or whatever they are binding to. And also, as in the DNA, we can determine the species they are coming from by those alignments. So, why are two sequences similar? This is what I was telling you before, that they might be reds of the same original molecule that we have cut into several fragments of different lengths and different coverings in the whole genome sequence. Okay, you can very well see it here. Okay, you can see the genome. That will be, this is a very silly example, but imagine that we have this quite short genome, and then to sequentiate it, we make cuts in it, and we obtain those reads, and then we have to align them and see which is the corresponding entire sequence. Okay. This is a very special case of alignment that we are not going to deep in those, but just you know that it's not the same. Okay, when we are aligning for sequencing or when we are studying the sequence itself. The other reason why two sequences can be very similar is the one that we already told, that they are homologous. They come from the same common ancestor. Okay, for example, Homo sapiens and this Pantoglodytes that I think is the chimpanzee. If I'm not wrong, this is the systematic taxonomy name for chimpanzee. And you can see that the alignment is almost the same, but this amino acid here that is not matching, but the rest of the amino acids are all the same. Another concept that you have to keep in mind is exactly what we understand in biology by homologous and analogous. Homologous, we call those proteins, for example, or parts of the body, of the anatomy, that are similar because of their origin, because of where they come from. And analogous is another term that we use to refer to those parts of the anatomy, for example, that are very similar in function, as you can see the wings here of the butterfly and in the bat, but they are not similar in their origin. Okay, they come from very different morphological parts of the original ancestor. Another concept that I know that there are a lot of concepts, but that you have a general vision of what this is, okay? And that you have to know is what is genetic variation. Genetic variation is the difference in DNA sequences between individuals within a population, okay? What we were talking before of variability. Variation can be seen as mutations or recombinations. What we want when we do sequence alignment is to omit, let's say, these ones or identify them. And these ones, we also find them, but as recombinations occur between long fragments, not just a nucleotide, but long fragments, and they exchange the whole fragment, it will be very, very easy to identify, okay? The complicated thing and the thing that we see most in alignments is this one, because when we align, we normally previously select those sequences that are more or less similar to the one we have. So recombination would be a part of our sequence that is in another place, but that is there and it is just the same. Okay, we have different variations in a sequence that can be classified from different perspectives, okay? The ones that we talk most in sequence alignment are derived from the effect those variations have on the genomic structure. We have substitutions, that is just a mutation that exchanges one base for another one. In this case, for example, you can see DA
Can
has. Yep.
Can I ask something? I'm just a little bit lost, like of the number of letter we need to say like it's a gene, it's not a gene, or how we say that starts here and that end here. I don't really understand that.
Can you repeat, please?
Like in insertion, we have something with 6 letter, and after that we have an insertion, so that is long,
It
more
has to
longer.
be. Yeah.
But I don't understand. Like, okay, there is more thing, but what that change? Like, it's
The number
longer.
of you are asking me for the number of nucleotides to consider something
Yes.
an insertion or a recombination, right?
Yes.
Yeah, recombination would be entire genes, for example, maybe 5,000 of base pairs, okay? So insertions will be maybe 10 nucleotides or maybe more. It can be more, but recombinations you will detect it because it is like an entire gene, okay? Because it's a fragment of the chromosome. When they recombine chromosomes. So it would be of the category of hundreds or thousands most of the times. Okay?
Okay.
Yep. Anything else? So I will continue. Yeah. And then insertions that we are saying that are mutations in which we have extra base pairs that are inserted into a new place in the DNA. That another thing that you have to consider is that it will be inside the gene, in the middle of the gene, for example. Okay. Also, deletions are mutations in which a section of DNA is lost or deleted. The later two, these ones, will result in what we call gaps when we align. That is those, imagine that you are aligning two sequences, we will see it later, but those fragments that has been inserted or deleted, when we compare with the other sequence, we will have an empty space. That is what we call gap. Also, another thing that comes from here is that some insertion and deletions may not be significantly affecting the structure of a protein. If we, for example, insert or delete a domain that is maybe those loop structures in a protein that are just to connect some kind of important domains, if we extract that or insert that, won't be very relevant for the function of the protein. So they don't always have the same relevance. Also, depending on the effect that we have on the protein sequence, we have different kinds of variations that comes from those in the slide before. Will, depending on how they are settled, they will be one or another. Okay, we have frame shift mutations that those mutations caused by the insertion or the deletion of any number of nucleotides that is not how do you say English? Divisible by 3. Do you remember the codons that we have different frame shifts from which we can translate those sequence of RNA to proteins. So if we insert a number that is not multiple of 3, we will change the rest of the sequence frames, leading to completely different amino acids. So the function would be completely different, okay? And we can have, for example, a stop codon a lot before than we would expect it, and we wouldn't have that protein. So we are losing that function of the protein, and maybe if it is quite essential, we have a problem there, okay? Also, we have synonymous mutations that are mutations that don't exchange the amino acid sequence. For example, as you saw the previous day, we have the genetic code that different codons may lead to the same amino acid. If we have a mutation, but that mutation leads to the same codon, it doesn't matter because finally we obtain the same amino acid, okay? And inside non-synonymous mutations, we can have neutral mutations, that is when the obtained amino acids is very similar to the previous one. They are in the same group, for example, of chemical structures, so the function of the protein will be the same. Or we can have a nonsense mutation, that is why I was telling you of those stop codons that lead to not having the protein, because it is not, we don't have the enough number of amino acids to codify that protein. Also, this is, instead of naming it protein function, we say gene function, because it's a thought of the point of view that we have another product or we have less products, okay? When we talk about loss of function, we understand it by the gene product that we will obtain has less or non-function, okay? Also, with gain of function is the opposite, that the gene product has increased or has a new function. For lethal, mutations that lead to the death of the organisms are for reversion, when we have a mutation and then we have another mutation that leads to the original sequence, okay? And then finally, variations that are classified for their effect on the fitness. The fitness is just a biological concept that defines the number of probable descendants that an organism has during his life, okay? How well is going to survive or evolve that organism in an environment. From that point of view, mutations can be deleterious, that is a mutation that increases the fitness of an organism, that decreases the fitness of an organism, beneficial, that increases that fitness, and neutral, that has no effect. The thing here is that the important variations that we are going to see today are these ones, okay? These ones are the ones that you have to keep in mind always, the substitutions, the insertions, and the deletions that give different types of alignments. I already mentioned that, but homology and similarity are not the same concepts. Homology requires similarity because of origin. Two things can be similar due to other reasons. Also, another concept is that homologous genes, those that come from an original common ancestor, can be also orthologous or paralogous. In this picture you can see very well. Paralogous genes are those that have been coming from an event of duplication. And then paralogous are those genes that would be obtained from a speciation event. Okay, so paralogous genes usually are not as similar as orthologous genes. This one would be homologous genes from globins that can be seen in mouses and in humans, for example. And they are all quite, well, these ones are all in humans, but as you can see, they lead to quite the same genes. They are, as they are in the same species, they will be, they will be orthologous. No, they will be paralogous. Okay, they come from an event of duplication in another species. We will say they will be orthologous. Here you can see better with the histones. We have an ancestral histone that has been duplicated in histone 1.1 and in another histone that is 1.2. Then we have an event of speciation that lead us to 4 histones, one H1.1 and in humans and 1.1 in chimpanzees, and then 1.2 in humans and 1.2 in chimpanzees. Okay, here you can see the one in the same species are paralogs, and the same gene in different species is called orthologous. Now we are going to start what we want to see today, the alignments. Okay, as you can see in the first example here, we have 2 chains, 2 sequences of DNA that we want to align. At first, if we just align them by the first nucleotide, you can see that only 3 pair bases are base pairs are aligned. But if we move one string to the right and the other one to the left, we almost align every nucleotide. Okay, so the first step that in biology we usually take to compare 2 sequences is to align them. There can be substitutions or changes in one residue with another, or gaps, as you can see. Here we have a gap and here we have a gap. Gaps, as I told you, are those missing residues that can be due to a deletion or to an insertion, if we are talking to the original sequence or to the other sequence what we are aligning to. You can see that gaps can complicate the alignments. It would be very easy to just align 2 sequences but by the first nucleotide, and that's all. But considering gaps, for example, here we have one and here we have another, can make that computation task very difficult, because we could have gaps wherever. So algorithms have to take into account that possibility of introducing gaps, and once we allow them to create those gaps, several alignments can be constructed between the 2 sequences, just not one alignment, but multiple alignments considering those gaps. Here we have also the globins, in where the picture in the top here, this one, represent what is the prototypical structure of a globin. Okay, and then in the bottom, what you can see is the the part of a multiple alignment between 8 globins. You can see that all these slabs represent those gaps, and yeah, at the end all of them will be obtaining this kind of protein. So even though they are not all exactly the same, they all are finally traducing the same function, translated the same function. Here, what we were talking, okay, that if we have this original alignment, if we don't consider gaps, we have different 5 different alignments. But as we have to consider gaps because there are insertions and deletions, the alignment gets really, really hard. You can see that here we have 5 and here we have 13 to pose a number, but they will be thousands of more alignments, okay, considering every position and every possibility with the gaps.
Do you use like metrics like Euclidean distances and something like that to choose the best arguments?
Wait, I can't see you. Ah, no, sorry. Can you repeat me the name of the metric?
Yes, like Euclidean distance.
Euclidean distance is not the one mostly used, but you could. We will be seeing now. But do you know Hamming distance or Levenstein distance, for example?
Levenstein, yes.
Levenstein. Those are most frequently used with sequence because Euclidean distance it's not seeing all of the possibilities when modifying one sequence to the other.
Okay.
Okay. Yeah, the same thing that we were telling that with no gaps. Here we would have 10 matches and that would be all. But if we insert just one gap, we would have 14 matches. And with 2 gaps, we have 16 matches. So it is the similarity increases when we insert gaps, but we cannot insert all gaps. We have to have some kinds of constraint that, okay? So first, from explaining what the algorithm of alignment does, I would like to say that we have different ways to visualize an alignment. This is what we call dot plots, in which we represent in one axis the sequence and in the other sequence. So here we will have, for example, sequence 1 and sequence 2. And we represent an X, what in visualization we represent with a dot, and that is why they are called dot plots. Whenever the nucleotides or amino acids are the same. This is just a way to visualize alignments, but we cannot have a metric to say this alignment is better or this alignment is worse with dot plots. But some relevant information can be obtained from them. The most important thing is that those regions that are similar, that are the same, would be represented as diagonals in the plot, and also substitutions appear as missing dots within a diagonal or not in each place. Okay. No, but substitutions are in the diagonal. Also, we have the gaps when we have displacements in the vertical or in the horizontal positions. This is what would be interpreted from a dot plot. If we had this dot plot and this alignment, here I remind you that 100, 200, 300 represent the number of pair bases, okay? Or if we had a huge sequence, it would be kilobases with KB, but normally in alignments that are smaller, we say BP, that is for base pairs, okay? So here in this sequence, sequence A, we align it to sequence B, and the nucleotide number 100 alineates to the number 1 of sequence B, and they match in those 100 pair of bases. In the dot plot, what we would see is a diagonal from the nucleotide number 100 in the sequence A to the nucleotide number 1 in the sequence B. So you understand, I mean, it is that diagonal from the start to the end of one sequence to the other sequence. Sequence B versus sequence A. Also, we can detect big insertions, deletions, and forward and reverse repeats by means of these dot plots. Here, keep it careful because it is represented just the opposite as we did in this matrix, because in this matrix we represented sequence A versus sequence B, but in those axes are represented in the bottom, not in the top, okay? So the diagonal is just reversed to the previous one. Here you can see duplications when you have two diagonals at the same time, or for example inversions when we have the diagonal the other way around. So it is cross to the original alignment. Okay. This is what would be a dot plot in real. Okay, not just those pictures that are very well seen. We can do a genome-wide analysis, what we call GWAS, with this kind of representations. GWAS is just an observational study of genome-wide set of genetic variants in different individuals to see if any variant is associated to a trait or phenotype or disease or whatever, okay? What we were saying about SNPs, for example. One limitation of the dot plot is that although we detect similar regions, we don't obtain the alignment. We don't see the two sequences aligned and which pair of bases are the same and which have been substituted, inserted, or deleted. So when we make alignments, we ask ourselves 3 questions. What do we want to align? How do we score an alignment? More or less what we were talking about, those metrics to search the similarity between those two sequences, for example. And how we define which is the best alignment, because as you saw, there are many options to align. So one of the critical points is which alignment is better than the other. The ones that provide us a higher similarity, probably, but we don't want neither too many gaps, neither too many insertions or deletions. So it is not as easy as it can be seen at the first part. time. So starting for what do we align? There are two types of alignment. What we call global alignments and local alignments. The global alignment algorithms try to create an alignment that covers completely both sequences, adding whatever gaps necessary, okay? In the other hand, the local alignments, those algorithms try to align only the most similar regions, although if they remove a region from one end of a sequence, because it can improve the alignment score when we are local aligning, okay? So local alignments are usually the best option unless we are sure that the two sequences we are comparing are similar, very similar. Besides, the local alignment algorithms will create a global alignment with both sequences covered if they are similar enough. Here you can see an example very easily of what are global and local alignments. Okay. Here, as you can see, global align the whole sequence, and local only just a part of those sequences. That would be this one here. Okay, you can see this one here. Uy, these ones are duplicated. Okay. Moving to the next part, how do we score an alignment? There are multiple ways of doing this score, of giving this metric of similarity to the alignment, this global metric to the whole alignment. Okay. One quite simple way would be what we were talking of distance as Hamming distance or Levenstein distance. Hamming distance is just the number of mismatches between the two sequences. So it is a more naive approach than those that we are going to see now. And Levenstein, considering two chains that don't have to be necessarily the same length, they can be different. Levenstein measures the number of editing operations that are necessary to convert one chain into the other. But what we usually do for measuring the score in sequence alignment is to have an scoring schema, okay? It's like if we do a scoring system that gives more points to alignments that are biologically more reasonable, because Hamming distance and Levenstein distance are for text mining or those kind of things that do not have assumptions below them, okay? So here we have some knowledge, biological knowledge, that we can apply to put better those scores, okay? So ideally, we would create a scoring system that gives more points to the alignment that align the homologous positions. Naive scoring systems would be to count the number of matching positions or the number of matching positions, for example, along a particular number of residues, let's say 100, for example. Usually the scoring systems also takes into account the number of gaps, and they penalize the alignment depending on the number and the length of the gaps that are present. So the main features taken into account to create a scoring system would be the number of matching residues, the one you can see at first point, the number of mismatches, the number of gaps and the length of those gaps. We can give different scores, like let's say sub scores, like we will then sum all of them, but sub scores to the different operations, matches, gaps elongation, and multiply them for the number of times that we have that event, okay? You have also to understand that one alignment can have different scores under different scoring schemes. So it will affect a lot the scoring schema that we select when doing an alignment. Once we have decided which schema we are going to use, the alignment algorithm should try to create the alignment that obtains the maximum score under the particular scoring schema that we have chosen. Every software that implements these kind of algorithms will usually have some default values for their parameters, okay? But these default values always have to be calibrated for the conditions we are studying. It depends, for example, if we are studying some codifying regions or non-codifying regions or which organisms, okay, whatever. Today we are not going to deep in that, but keep it in mind, okay? And here, the score of an alignment, as I told you, is the sum of the scores for pairs of alignment characters plus the score of gaps. What we were saying that we would have to define which score we are going to give for mismatches, which for gaps creation, and which for gaps elongation. For example, for the alignment you can see here, we will sum the score that we would give to the alignment of the amino acid valine to alanine. We would have had a score for that alignment because they are quite similar, or they are not quite similar, whatever. In this case, because they are quite similar, okay? Then the one of A to E, from H to Q, and then etc. And for gaps. Here we have 3 gaps, we have to sum the 3 gaps, and then here we have 2 gaps, we have to sum the 2 gaps. The gaps will be penalizing the more, okay? So we can divide to think this in a proper way in 2 steps. One, we need to have a gap penalty function that will indicate the ghost of a gap of length k, and then we need to previously determine the substitution matrix, that is a matrix that indicates all the amino acids versus all the amino acids, which is the score of substituting one amino acid for the other. There are different methods to estimate that, okay? And also we can use some heuristic methods that are a little bit quicker, although they won't obtain the best surely alignment, but they provide us a good solution, and is what we will be doing in the practice today. Okay, for gap penalty functions, the most basic one is a linear gap penalty functions. Okay, when we have different gap penalty functions that we require different dynamic programming algorithms. We will see it later. The simplest case is when a linear gap function is used. So we penalize the gaps with a k number. For example, we say that gaps penalize 10, for example, and we have three gaps, so 3 multiplied by 10, 30, okay? Here, there is a confusion. Here would be k. K is a constant. Gaps, g would be the number of gaps. Okay, that was the idea. I will change it later. So the substitution matrices are popular for when we are aligning protein sequences, okay? Because they are very well studied and they are obtained from evolution and phylogenetic studies. So it is demonstrated that these both have a biological reason below. They try to capture the relative substitutability, so the capacity of substitutions of amino acid pairs in the context of evolutions. Here, for example, is one of the matrices that is PAM. You can see all the amino acids and all the amino acids, and which would be the cost of converting one to the other. If those amino acids are quite the same, the cost will be very, very low. But if one amino acid is for one thing and another one is chemically very, very different, the cost will be very, very high. Here is another one that is Blossom. Just for you to know that when we are measuring the alignment, those score how they are obtained, but you don't have to really know much about this. Yeah, they are different also, PAM and Blossom matrices, depending on the number of mutations or in the percent of identity between the amino acids. Okay, it doesn't make much. It's not very relevant for you. What it is relevant is these heuristic methods that we were talking, as BLAST, for example, that we will implement it in a couple of minutes. The previous algorithms that we have seen take too much time to align sequences, and if we want to search in large databases for some sequences, we need other methods that help us approximating those functions. Okay. So we will see BLAST now, but before that, the third question that we had, it was how can we define which is the best alignment? This is normally used by dynamic programming, that I'm sure that you will know more of dynamic programming than I do, that it is to solve an instance of a problem by taking the advantage of the solutions of different subparts of that problem. So we can reduce the problem of finding the best alignment to find the best alignment of all of the prefixes of a sequence. So we can avoid recalculating the scores already considered. There are two main algorithms that are quite important for this kind of programming, that are the Niemann-Bunz and Smith-Waterman. The first one is for global alignment and the second one for local, but they more or less do the same thing. You have to think that when we have to align two sequences of, for example, 100 base pairs. That is not a lot. It's just a small sequence of DNA. We have up to 10 elevated to 77 possible alignments. So it's like crazy. We need something to keep it that simple, okay? But yeah. What this kind of algorithm do is to, from the two sequence and a scoring schema that we have defined, they guarantee to get the alignment with the highest scores, but also the memory and the time that this scores is high, is of the order of the sequence length of one sequence and the length of the other sequence. And the results will depend on the schema that we are using for the scoring. Okay. Here you can see what the Needleman Bones does. That is keeping all the scores from, for example, using the Blossom or the Palm matrices, substitution matrices. They go searching which are the smallest scores, and they keep that path, let's say. But this is easy for those little sequences. But when we have to search and make multiple alignments, or we have to search for a concrete particular sequence in more databases, is not as easy. Okay, so for this we have BLAST. BLAST, I don't know if it does ring you the bell, but it is the most cited paper in the history of science. The one that was explaining this algorithm. It stands for Basic Local Alignment Search Tool, and it is like an application most than an alignment method itself. Okay. If we have a query sentence with BLAST, we can look for similar sequences inside a database. As I am saying, BLAST is one of the most used algorithms in bioinformatics, and it includes a family of related algorithms capable of aligning a query sequence against all the sequence in a database in a very fast way. That is what is quite good, that we can find our sequence what is matching to in a huge database. So BLAST will produce local alignments of the query sequences against the sequences in the database. It will also generate an expected value, what we call E-value, that is related with the statistical significance of that alignment. Is the way that it has to say that we have the best alignment result, because as we don't are using that dynamic programming that will guarantee us that we are obtaining the best alignment, we have to search other ways to get the best results. Okay, so this BLAST is implemented in the NCBY web page and in their servers, and also in MBL, I think. We can also get our software version of BLAST, and if I'm not wrong, there is a Bash script for doing BLAST. Also, I think Python has some version of BLAST. It is, as I said, the most used bioinformatic tools, because it is used for searching in the databases. We use it for to see our sequence, which species come from, or also to annotate the function of that sequence. If we know the sequence we have is related because it aligns very well to another sequence, and that sequence has a protein that its function is blah blah blah, we could take that blah 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 to our sequence. Also, we can detect which family is that sequence from, or study the phylogenetical relationships with other species. This is the same, that how could we look for a similar sequence inside a big database? Those are the sequence only from GenBank, the number of sequences and the number of residues. I mean, the numbers are huge, so we have to have a way to optimize in that. To do BLAST, we have to search for words of the query sequences that are generated, and those words are looked in the database indexed. The key here is that the database will be indexed in different subject sequences. So when we are looking for our query sequences, we'll have that structure of indexes, and it will be more quickly, okay? Yeah, as I said, we have to establish an statistical threshold to decide whether our alignment is significant or not. And this process will be very fast because of that, because we have indexing algorithms in the search. So yes, two steps. With our query sequences, we make alignments with the subject sequences in the database. Once we have those alignments, we study the significance, and we just return those that have any value upper to one that we want. I remind you that query is how we call our sequence, subject the sequence that are in the database, hit or match are those subject sequences that have a similarity with our query, and HSP is one of the alignments between a query or a subject. Exactly what it means, HSP is high scoring pair. So the steps BLAST algorithm will take would be splitting the sequence that we have, our query, in different fragments that we call words, looking for those words in the indexed database, that as we have indexes, it would be a lot more quick here. Then we will retrieve the subject sequences with matching words, and we will do a fast and approximate alignments with every matching subjects. Okay, as you can see, we have all of these subjects in the database. We just get those that can align, and then we study the different alignments, and finally we estimate that significance. You can see here another representation of what we would be doing, okay? So the advantages of BLAST are that it is quite fast, and that tries to align both the forward and the reverse strand. So if our strand is by chance, for example, inversed because of some process or recombination or whatever, we would also find those sequences that are aligned with the reverse strand, okay, the one that goes 3 to 5 instead of 5 to 3. It is very convenient because it is available in the web, and it also does a dot plot along with the test alignment. The limitation is that it's not as sensible as Smither-Waterman, the one that we saw before, that is more precise and always returns one of the alignments with the highest score. Okay, always the one with the highest. Also, BLAST can fail to detect the similarity if there are too many differences between the species, and a very distant sequence won't be detected. The smaller the words are, the more sensitive they are, but they require larger databases. And usually, although it's not a big problem, it can cost a little bit of sensitivity. With BLAST, as we have been saying, we have not a guarantee to get the alignment with the maximum score. So BLAST usually generates several HSPs between the query and one subject sequence. And there are also another type of BLAST that are derived from BLAST, that could be MegaBLAST, that it is a little bit faster than BLAST, although it's also a little bit sensitive. It will miss the match if the sequences are very different, okay? And also, depending on the kind of sequence that we have, there are different programs that compare different kind of sequences. We have BLAST-N that are query nucleotides, and the subjects are nucleotides databases also. BLAST-P, where the queries are proteins, and the subject databases are also proteins. BLAST-X, where the queries are nucleotides, and the databases in where we look are proteins. Okay. T-BLAST-M, where we have a query of protein and database of nucleotides, and T-BLAST-X, where we have nucleotides as queries and database of nucleotides, but we have to translate everything, so it would be like this, okay? Here, protein-protein, DNA-DNA, DNA to protein, protein to DNA, DNA to DNA, but translating everything. They are different, and we would use ones or other depending on different things. So, the problem of BLAST is that even if we have random sequences, we will have a best scoring alignment. Even we invented the sequence and it is a random sequence, with another one it will also align, because of the we only have 4 pairs of nucleotides, so in some way they will align. So how could we know which alignments are statistically significant? So we make a distribution, we create all the alignments between the random sequences, and create a distribution of the scores for alignments between random sequences. And once we have this, we see where the random are, where the similar score to alignment between random sequence stops, to say it somehow, and we only kept those, for example, 0.05 larger, higher score that are not likely to be due to random similarity, what we call, for example, a p-value, but this one we will call e-value because it's not a probability, but a probability multiplied by the number of sequences in the database. So we don't have a 0 to 1, but 0 to a large number. Inverse, okay? So the expected value is the number of hits between random and related sequences that one would expect to see with the given score. Now, which time is it? Okay, perfect. I want you to go here to this page. I will put it in Moodle so you can just copy paste and not One sec. Here. So you are here, right?
Yes.
Perfect. We are going to do the easiest one, that is nucleotide to nucleotide. That would be BLAST N, it is called. And here we have it in the first box. We click in it. And here is where we do BLAST. There are different parameters and different things that we have to fulfill. The first one is the query that we are going to study. We could just enter the accession number if we knew it from the NCBY, or enter the FASTA here, copying and pasting, for example, the one that I give you. But as we have the file, we can just upload it from the file. So I have it here, and I will upload it. The one that we were working with before, that was the one of the rat that just had one sequence. It was not a multi-FASTA. I remind you things. Because it only had one major sign, and we open it, okay? And now we have it here, you see. Oops, sorry. We also could determine a subrange in that sequence to just work with some substring of that complete sequence, but it's not our case. We want the whole gene. And here we can give a title for the job, so we can recognize the server, but if you don't enter anything, it just assigns you a number and it's all okay. Then we have to choose the collection of databases that we want, okay? Because I remind you that for BLAST we need a query, a database set where we will be looking for the subjects, and then the method that we will be using for the BLAST, that can be nucleotide to nucleotide, nucleotide to protein, etc. Okay, so here I'm going to search for standard databases. For example, that we could be looking for RNAs. Not my case because I am entering DNA. Genomic and transcript databases, not my case because I'm just entering a little gene. And beta coronavirus, I'm pretty sure they have added this lately because I didn't see this before. Organism, we want to search, we know that we have a rat gene here, okay? But what I wanted to analyze is if that sequence that we have sequenced from other organisms, we can relate it to any sequence in humans, okay? So I'm going to enter for organism Homo sapiens, that is humans. Homo sapiens, taxid 9606, okay? We could also enter the ID for the NCB interest query that would give us the particular genome, for example, or a set of databases that we would like to use, but with this for us is fine. Okay. Here we can choose MegaBLAST or other type of BLAST, but I would choose MegaBLAST because I remind you that if we have quite similar sequences, MegaBLAST will work well and it will be faster than BLAST itself. so it's fine for us. I will mark to show the results in a new window, and for the algorithm parameters I won't touch anything, but just for you to know that those are the things that we were talking that we can define. For example, the expected value threshold, that it is always settled for 0.05. But if we would like to change it, we could. Also, for example, the word size, the word that we are dividing the sequence when we are searching for it in the different subjects, or the maximum number of subjects that we are targeting, for example. Those parameters that are set like that because of something. So I am not going to change it, but you can also, for example, change the function of the gap penalty, a lot of things. I'm going to start running it, and it will take like 2 minutes or so. What? Oh, I didn't. What happened?
I think you didn't load the file.
I didn't load the file when I clicked it.
Yes.
Yeah, okay, thanks. Perfect. So yeah, now it is working. Are you all in the same point as I am? Or anyone has any doubt or something?
No.
Okay,
No.
it's easy. I mean, it's not very much. If you don't have to change very much the parameters, it's just inserting the query sequence, identifying which ones you want to use as databases, and that's all. Okay, now we have here the results. We can also obtain the graphic summary, the different alignments, the taxonomies. The most similar sequence that it has obtained is the first one. We can we can, how do you say, order by the different metrics that is giving us. For example, the identity, they have the two sequences. This one would be much more similar, but as the e-value is larger because the other one were 0, we are not so sure that this hasn't been by random. Okay. If we, for example, choose the one that we have first before, that is this one. My I don't know if it is my internet or if it is their servers that are a little bit slow. If I open this, we can see the. Wait. Okay, we can see if we choose one of those, we can see the specific alignment that it has made. Okay. The identities is the percentage of similarity between one sequence and the other. It gives us the percentage of gaps also. A lot of relative relevant information. Also, if we click in graphic summary, we can see the best alignments it has made. Also, if we click in the first one, for example, it redirects us to the same alignment that we were seeing before. Wait, where is my mouse not working? Oh my god, doesn't matter. Okay, so yeah, that's pretty much all. The thing that I want you to have clear from this is, if, for example, I know that some gene is causing, some mutation in a gene is causing a disease in mouse, I would look for those mutated sequences in humans and see the relationships between the genes in one species and another, or if we have other species in which we can study, because in humans it is very difficult to study experimentally, and we would need some animals or more model organisms. It could be, I don't know, the Drosophila, for example, but as it doesn't have much similarity to human, it isn't as reliable as, for example, mouse that is a mammal, okay? So here also you can see the taxonomy of the organisms that we are searching for, okay? What time is it? 10 to... Let me check how much do we have left, so I decide if we continue the 10 minutes that it rest of. Okay, no, we have just to see multiple sequence alignment. That is when we want to study different regions in different sequences to see how conserved they are, or the similarities between different species. For example, we align more than two sequences, what we were doing now, that are pairwise alignment, like 2 by 2. And other types of algorithms and methods are used, but I think we will see it the next day. Any doubt till here, or something that you are curious?
No, it's clear.
No.
Yeah? Okay, so yeah, it's quite easy, okay? I'm not going to ask you in the practice things very that you have to have a biological background, let's say, because the stuff here is that sometimes when we are analyzing sequences, we have to have a clear idea of the processes behind those sequences. And for that, normally what we have to do is research in the literature or something else, and then we can define exactly what would be our best parameters or algorithms to use. So yes, don't worry about the practice, because it would be very, very easy, I promise. And next day we will see multiple sequence alignments, and I don't think we will stop much in bioinformatics or biological data concepts. I think the next day would be more useful for you to have a clear vision of diseases, codifications, classification systems, taxonomies, more orientated to human and diseases, because the thing of alignments is that most of the times are used for comparing species and not just humans. A lot of times it's relevant for us compared to other species, but for diseases, most times we just focus on humans or in mammals or some model organisms that are quite related to us, okay? So, yep, I think that's all for today.
Thank you. That was a great class.
Yeah, thank you.
Yeah. Thank you.
Thank you. See you.
See you. Have a good day.
Have a good day. Bye.
Thank you.